of meiosis 1 is that every single one of your chromosomes needs to be replicated. So now that you've drawn all those, make them all look like X's. Because each individual strand, moms and dads, has to all replicate. And at the end of this step, we'll get 46 X's in that cell, okay? So that was interphase 1, prophase 1. Now we have to move on to metaphase 1, okay? Mom and dad's chromosomes line up on the metaphase plate. 23 X's on one side, 23 X's on the other. You can do this in any order because it's very random. So I have some pink and blue over here, and I have some pink and blue over here. Just make sure your total pinks and total blues add up to 23 each. In anaphase 1 of meiosis, this half splits from the other half, and they end up in 2 new nuclei. So my left side of my metaphase plate here, those chromosomes end up in 1 nucleus of 1 cell. And the right side of my metaphase plate, these 23 chromosomes end up over here, okay, in a second cell, all right? Notice in each of my 2 new cells, okay, there's a mix of both mom and dad's chromosomes, but the total number is still going to be 23, okay? So since these are 2 new cells, each with 23 X's, okay, we're going to move into meiosis 2. Each of these cells is now going to start meiosis 2, which looks exactly like mitosis. So my X's line up on the metaphase plate. 23 here, 23 here, okay? The X is going to get pulled apart in either side. So half the X to one side, half of the X to the other. They're going to go back to looking like they did originally before I replicated them. I now have 4 cells with 23 chromosomes, a mix of both mom and dad's DNA, okay? And since I said that we were working in an ovary, I made 4 egg cells. If I was working in the testicles, I would get 4 sperm cells made. The exact same way. The same thing happens, but the result would be 4 sperm instead.